Imagine a wire,
Yeah.
right? But it's no thicker than a human cell.
Barely visible, even under standard microscopes.
Exactly.
Right.
And it conducts electricity better than traditional biological materials. You can literally boil it in pure acid, and if you snap it in half, it simply regrows itself.
Which is wild on its own.
It really is. But here's the kicker for you listening today. It's core architecture. It's made from the exact same structural blueprint as the protein responsible for mad cow disease.
Yeah, that's the part that usually makes people pause.
Right. So today we are taking a deep dive into something that essentially bypasses the entire Industrial Revolution. We are leaving behind like the era of mining steel and smelting copper.
Yeah.
We're unpacking the source material that completely rewrites how we build a civilization.
It's a profound shift. I mean, we are looking at a future where you don't build infrastructure anymore. You grow it.
You just grow it.
Yeah. And the most shocking part of this whole thing is that the fundamental building...
Looking at a comprehensive, multi-part theoretical framework. It's by an independent researcher, Zachary David Enfield.
And it spans a lot of ground.
It really does. It starts from this foundational paper titled Prion and Amyloid Architectures, a total application framework. And then it goes all the way through these incredibly dense, fascinating theoretical addendums.
Right. It covers everything. Nuclear energy conversion, civilizational geometry, and something called thermodynamic computing.
Which we will absolutely get into, but I know the scope of this sounds like absolute science fiction.
Oh, totally. Which is why it's vital to ground this right here, right now.
Yeah.
Everything we are going to explore today, like every physical mechanism, every material property, every strange quantum effect we talk about, it's all drawn from peer-reviewed experimental literature.
Real science.
Exactly. Published between 2003 and 2025. The synthesis of these ideas is radical, sure, but the physics and the chemistry, they are fully documented facts.
Okay, let's unpack this. Our mission today is to understand what's called the cross-beta architecture. We need to figure out how you take a microscopic biological fold and use it to replace, steel, silicon, and lithium-ion batteries.
Right. Because before we can build a galaxy-spanning civilization, we have to understand the brick.
Exactly. The brick. So what exactly are we looking at when we say cross-beta?
So to visualize this, you have to imagine a single microscopic thread. In the literature, we call this a fibril.
Okay, a microscopic thread.
Right. And in a cross-beta architecture, you have these protein strands. They're specifically called beta strands, and they are oriented completely perpendicular to the long axis of that thread.
Perpendicular. So wait, imagine a ladder where the rungs are the beta strands.
Yes, exactly like a ladder.
Okay, I'm picturing the ladder.
Now imagine those rungs are stacked incredibly tightly together. They're bonded by these things called hydrogen bonds, forming these super dense sheets that run parallel to the axis of the thread. But what makes this a literal masterclass in materials science is that it naturally forms a nanocomposite.
A nanocomposite, meaning it's made of different parts working together.
Yeah. You have these alternating layers. One layer is the highly rigid hydrogen-bonded beta sheet we just talked about, but then extending out from them, you have the protein's side chains.
And what do the side chains do in this ladder?
They essentially act as molecular shock absorbers.
Oh, wow.
Yeah. Because think about a standard rigid crystal, like glass or even certain metals. If you hit it hard enough, the bonds just snap.
Right, it shatters.
Exactly. But in the cross-beta architecture, the energy of a physical impact is transferred into those side chains. They flex, they dissipate the kinetic energy, and they protect that super rigid core.
So it's almost like microscopic macro, like, the material on the inside of an abalone shell.
Yes, that's a great comparison.
It's incredibly hard, but also flexible. Or like human tendon, but hold on, I have to stop you here. Because you mentioned this is the architecture of prions. We are talking about building our cities, our computers, our physical world out of the exact same biological mechanism that causes fatal, untreatable brain diseases.
Right.
I mean, that sounds like a biological apocalypse waiting to happen.
It is honestly the first most glaring question anyone asks when they look at this research. And Enfield's framework draws a very hard, non-negotiable line right here.
Okay, good.
We have to separate the physics of the fold from the mammalian disease. The proteins used in these engineering applications are absolutely not the mammalian prion proteins.
So they don't cause mad cow.
No, they don't interact with human biology in that way at all. They are derived from yeast prion determinants, like the SUP35 protein, or they are entirely synthetic, lab-designed peptides.
Wait, yeast has prions. I didn't know that.
It does, but they function entirely differently. In yeast, prions are actually an evolutionary adaptation.
Really? Like a feature, not a bug.
Exactly. It's a way for the yeast to rapidly change its physical traits to survive environmental stress, and it does this without having to actually mutate its underlying DNA.
Oh, that is fascinating.
It is, but the crucial point for us is that a yeast prion cannot cross the species barrier to infect a human. The amino acid sequences are just completely incompatible.
Okay, so it's like trying to run an iOS app on a Commodore 64.
Perfect analogy. It just won't compute. The architecture of the self-assembly is what we want. We are literally just borrowing the geometric trick of how the protein folds and stacks without any of the biological hazard.
Okay, so we have isolated the physics from the pathology. We have a safe, self-assembling brick.
Yes.
But if we are talking about replacing steel, this microscopic thread has to have some unbelievable mechanical properties. Like, how tough is it actually?
The metrics are almost hard to believe until you actually read the experimental data. Engineered polymeric amyloid fibers have achieved a toughness of 160 megajoules per cubic meter.
Okay, I need an analogy for that. How does 160 megajoules compare to materials we know?
Well, it outright beats natural spider dragline silk.
Wait, really? Spider silk has been like the holy grail of ultra-tough biological fibers for decades.
It has been. But this beats it, and it achieves that extreme toughness at roughly 1/8 the density of steel.
1/8 the density.
Yeah. But honestly, raw strength isn't even its best feature.
What could be better than beating spider silk?
What makes it truly revolutionary is a concept the framework calls the hydrogen bond density dial, or HBD.
Okay, so we have a physical dial we can turn to change the material. How do you turn a dial on a microscopic protein?
By changing the sequence of the amino acids before it even folds, the mechanical properties of these fibrils are directly proportional to how densely packed those hydrogen bonds are in the core.
I see.
So if you want a structural steel analog, say, you're building a massive physical strut for a building, you tweak the genetic sequence to pack those residues extremely tight.
You turn the HBD dial all the way up.
Exactly. You maximize the HBD and the fibril becomes incredibly stiff and rigid.
And what if you turn the dial the other way?
If you introduce polar-rich cores and decrease the HBD, you get a highly dynamic, fragile fibril.
Fragile. Why would we want it to be fragile?
It sounds bad, I know. It easily fragments under stress. But you have to realize those fragments instantly become seeds to grow new fibers.
Oh, wow.
Yeah, you've essentially created a dynamic, self-replicating scaffolding.
That is wild. You can dial in the exact mechanical properties you need just by typing a different sequence into a gene synthesizer.
Exactly.
But what about wear and tear? I mean, steel rusts, concrete crumbles, biological things usually, rot.
This is where the cross-beta architecture just leaves traditional synthetics in the dust. The chemical stability profile is absurd.
How absurd.
First off, they're highly resistant to proteases. Those are the enzymes in nature that usually act like microscopic scissors to chop up proteins.
So natural decay doesn't really affect them?
Right. But it goes way beyond that. Certain synthetic amyloid sequences can actually survive being boiled in concentrated sodium hydroxide.
Wait, boiled?
Boiled. And they can survive total submersion in pure formic acid.
Stuff that would dissolve normal organic matter into literal sludge.
Exactly. It doesn't touch them. And most impressively, they survive massive doses of gamma-ray radiation.
Gamma rays?
Yeah.
Like space radiation?
Yeah. If you blast a normal silicon logic board with gamma rays, the radiation knocks the atoms right out of their rigid crystalline lattice. The chip just fries.
Right, because silicon is brittle at the atomic level.
Exactly. But because the amyloid fibril is held together by an immense network of dynamic, self-healing hydrogen bonds rather than brittle covalent bonds,
It just absorbs it.
the radiation either passes straight through or the bonds briefly break and then simply snap back into place.
Wow. Think about what this means for you tuning into this right now. If you're wondering why this matters to your life, imagine a material that is lighter than aluminum, tougher than Kevlar, completely biocompatible.
And biodegradable when we want it to be.
Right, but it can literally survive the blistering radiation of deep space and the most caustic chemicals on Earth.
It is the ultimate programmable matter.
It really is.
But as incredible as that is, a strong, indestructible brick is really just good for building a wall or, a spaceship hull. It's fundamentally passive.
Right, and a wall is boring compared to what comes next in this framework.
Absolutely.
Because what if that brick could conduct electricity? What if it could sense its environment or generate its own power? Here's where it gets really interesting for you guys listening. We are transitioning from dead structural scaffolding to what we might call thinking wires.
Yes, and this transition from structure to electronics, it begins with a landmark experiment back in 2003.
2003, so this has been around for a while.
Over 2 decades. It was by a researcher named Scheibel and his team. They took that harmless yeast prion we mentioned, SUP35, and induced it to self-assemble into a microscopic fiber about 10 nanometers wide.
Okay, a tiny protein thread.
But then they did something absolutely brilliant. They used that biological fiber as a template for selective gold deposition.
Oh, so they grew a protein thread, and that thread chemically guided free-floating gold atoms to stick to it, coating it perfectly.
Precisely. They created a flawless, continuous gold nanowire.
That's incredible.
Think about how we make computer chips today. It requires massive, multi-billion-dollar fabrication plants, extreme vacuum chambers, highly toxic chemicals, and complex photolithography just to etch tiny wires into silicon.
And it's incredibly resource intensive.
Exactly. But Scheibel proved you could just toss a handful of molecular seeds into a beaker of liquid and literally watch a highly precise electrical grid grow itself.
It's like tossing seeds into a garden and growing a power grid. But the framework goes even further than that, right? Because coating a protein in gold is cool, but it's still relying on a metal.
Yes. And that brings us to a massive 2021 discovery by Ships and colleagues. They demonstrated something called tyrosine hopping.
Okay, tyrosine hopping. I know tyrosine is an amino acid, like a basic building block of proteins. But how does it hop?
So tyrosine has a unique shape at the atomic level. It contains a carbon ring. And the researchers discovered that if you design your amyloid sequence to have a very high concentration of tyrosine, those rings stack up on top of each other as the fiber folds.
So they just align themselves.
Yeah, it creates a phenomenon known in organic chemistry as pipey stacking.
Pipey stacking. Let me see if I can picture this. So you have these microscopic rings, and they stack up like a roll of coins inside the core of the fiber.
That's a perfect analogy, a roll of coins. And because those rings are stacked so incredibly tightly, their electron clouds physically overlap.
Okay.
They create a continuous orbital pathway. Electrons can literally hop from ring to ring, traveling down the entire length of the fiber over micrometer distances.
Intrinsic conductivity.
Exactly.
You don't need the gold anymore. The biological fiber itself is the wire.
No metals required whatsoever. And it's not just electrons either. These engineered scaffolds have demonstrated proton conductivity that vastly surpasses synthetic biomaterials.
Really? Protons too.
Yeah, you can literally grow self-assembling proton exchange membranes for hydrogen fuel cells entirely out of a protein soup.
So the wire grows itself, and it conducts power.
Yes.
But this framework claims the material also generates power. It says these fibers are piezoelectric. And I think we need to really break down what piezoelectricity is for this to make sense for the listeners.
Absolutely. Piezoelectricity is a phenomenon where a material generates an electrical charge when you subject it to mechanical stress.
So applying pressure.
Basically, yeah. If you squeeze it, bend it, or vibrate it, it produces a voltage. At the atomic level, it happens in materials where the positive and negative charges are separated into an asymmetrical lattice.
Like they're slightly off balance.
Right. When you apply physical pressure, you force those charges even further out of balance, and that shift creates an electrical current.
And we use this all the time in daily life.
Right.
Like the spark igniter on a gas grill. You click the button, a tiny hammer hits a piezoelectric quartz crystal, and it generates a spark.
Exactly. The mechanical force becomes electricity. But in 2010, researchers discovered that these microscopic amyloid nanotubes exhibit massive piezoelectricity.
How massive?
We are talking about 60 picometers per volt.
Wow.
Yeah, that doesn't just match biological materials, that directly rivals heavy industrial ceramics like zinc oxide.
Wait, microscopic protein threads are generating as much electricity under pressure as industrial ceramics.
Yes. They are. And what's really fascinating here is how researchers amplified it using a principle called co-assembly enhancement.
Co-assembly enhancement, meaning building it together.
Exactly. They didn't just use one type of peptide, they mixed two different ones. For example, a diphenyline peptide and a phenylalanine tryptophan peptide.
Okay, two different building blocks.
Right. By forcing these two slightly different shapes to co-assemble into the exact same tube, they break the symmetry of the lattice even further.
Ah. So they made it even more asymmetrical, which makes the piezoelectric effect stronger when you squeeze it.
Precisely. They boosted the voltage output nearly threefold just by mixing them. And remember, the raw materials here are literally food grade.
Food grade.
You are building highly efficient electrical generators out of derivatives of lysozyme and insulin.
That is just wild. But I have to ask the obvious skeptical question here.
Go for it.
If this material is so incredibly sensitive that it generates electricity just from ambient vibrations or sound waves,
Yeah.
how do we stop it from constantly short-circuiting its own environment? I mean, if we embed this in living tissue, isn't it just randomly zapping everything around it?
It's a great question, but that sensitivity isn't a design flaw. It is actually the defining feature that makes this the ultimate biomedical tool.
Okay, how so?
The framework explains how this piezoelectric property makes the amyloid scaffold a wireless, battery-free therapeutic device.
How does that work in practice? Like, give me an example.
Let's use cancer treatment as an example. You inject these specific tissue-targeted piezoelectric amyloid fibers directly into a tumor.
Just right into the mass.
Right. And initially, they are completely inert. They do nothing. But then from completely outside the patient's body, you apply targeted high-frequency ultrasound directly at the tumor site.
Okay, so the sound waves hit the tumor, and they physically vibrate those tiny protein fibers.
Exactly. And because the fibers are highly piezoelectric, they transduce that mechanical acoustic energy from the ultrasound into a localized electrical charge.
A voltage spike inside the tumor.
Yes. That sudden spike in voltage drives chemical reactions in the immediate surrounding fluid. Specifically, it strips electrons away from local molecules, creating what are called reactive oxygen species, or ROS.
Reactive oxygen species, so it generates severe oxidative stress.
Precisely.
It's basically localized chemical warfare against the tumor, tearing apart the cancer cell walls from the inside.
Exactly. And you accomplished all of this with no batteries, no wires protruding through the skin, and no invasive power sources whatsoever.
The microscopic fiber acts as the transducer, the chemical catalyst, and the delivery vehicle all simultaneously.
It does everything. And when the treatment is done, you can actually engineer the sequence so that natural enzymes eventually break it down into completely harmless amino acids.
That is just phenomenal. Okay, so to recap where we are for everyone listening, we have an indestructible, self-powered, highly conductive material.
Yep.
And it grows exponentially. You drop a seed into a vat of raw amino acids, and it just relentlessly copies its own geometry. But unchecked exponential growth is basically just a tumor.
It's great goo.
Right. So how do we actually tell this material what shape to take? If I want a circuit board or a replacement heart valve, how do I stop it from just growing into a chaotic blob?
This is the great hurdle of biological manufacturing. And it brings us to the third section of Enfield's framework, prion optogenetic lithography.
Optogenetic lithography, that's a mouthful.
It is. But this is where we learn how to actually write solid space with light. It is the convergence of three totally distinct scientific pillars into a single technology.
Let's break down those three pillars, because this is where the framework goes from really interesting chemistry to actual science fiction made real.
Okay, the first pillar is the one we've been discussing, prion self-assembly.
Right.
That is the engine. It's the amplifier that physically grows the material out of the liquid. The second pillar is optogenetics.
Now, I know optogenetics is heavily used in neuroscience, right? Like to turn neurons on and off with lasers.
Exactly. At its core, optogenetics uses specific light-responsive proteins, things like Cry2 or LOV2.
Okay.
These proteins contain a chromophore. That's a molecule that specifically absorbs photons. When a photon of a very specific wavelength hits that chromophore, it literally causes a chemical double bond to flip into a single bond.
So it changes at the atomic level.
Yes, and the mystical shape of the entire protein twists and unfolds in response to that light.
And what's the third pillar?
Photolithography. This is the spatial control. It's the exact same basic principle we use to etch silicon wafers today.
Using light projectors.
Right, using a projector to cast highly structured, microscopic patterns of light into a specific 3D volume.
Okay, I see the pieces. But how do you combine a growing prion, a light-sensitive folding protein, and a projector?
You engineer a fusion protein. You literally take the synthetic prion sequence, the part that wants to stack into a tough fiber, and you physically fuse it to the light-responsive optogenetic protein.
You stick them together.
Exactly. You create a molecular lock and key. In the dark, the optogenetic domain is folded up tight, and it acts like a physical cage. It holds the prion sequence trapped in a shape that simply cannot self-assemble.
So if I have a giant vat of this stuff sitting in a completely dark room, it's just a liquid. just a harmless soup of floating locked proteins.
A completely inert liquid. But then you turn on the projector, you shine a highly precise three-dimensional pattern of light, let's say a specific frequency of blue light, directly into the center of that liquid vat.
And wherever the blue light touches.
The photons hit the optogenetic cages in that specific illuminated volume. The cages absorb the light, change their shape, and spring open.
Oh, wow.
The prion sequence is suddenly released. It snaps into its cross-beta geometry, and the self-assembly cascade instantly begins.
And I'm guessing this is the crucial part. It only nucleates and grows in the exact volume of space where the light is shining.
Precisely.
You are literally projecting a hologram into a vat of liquid, and a solid object materializes out of nowhere, matching the exact 3D shape of the light.
It's exactly like that.
It's like a 3D printer.
Yeah.
But you aren't awkwardly extruding hot plastic layer by layer, nozzle by nozzle. The entire object solidifies simultaneously volumetrically wherever the light touches.
The framework calls this the replicator paradigm. You are casting matter from light, and the applications outlined in the sources are just staggering.
Give us an example.
Take food production. Instead of the massive energy and land overhead of raising a cow just to grow muscle tissue, you project the microscopic architectural pattern of muscle fibers into a bioreactor full of nutritive protein.
So you're printing the steak.
The light triggers the cascade, and you grow a highly structured meat analog with absolute molecular precision.
Or medicine. Imagine you have a patient with severe spinal cord damage.
Yes.
You don't try to stitch severed nerves together. You project the exact geometrical shape of a neural pathway directly into the damaged tissue.
Exactly.
And you use light to grow a conductive piezoelectric scaffold right there in the living body, giving the patient's own neurons a physical bridge to regenerate across.
It is entirely in vivo fabrication.
But hold on, I have a massive mechanical problem with this.
Let's hear it.
You shine the light, the lock opens, the seed forms, and the runaway prion cascade starts. That all makes sense.
Right.
But what stops the cascade when you turn the light off? I mean, if the end of the fiber is exposed, doesn't it just act as a permanent seed? Doesn't it just keep recruiting more liquid protein and grow wildly out into the dark areas of the vat?
It's an excellent question. The problem of cascade length.
Yeah.
Because if you can't stop the growth, you don't have lithography. You just have a delayed tumor.
Right. So how do they stop it?
The framework details several engineered boundaries to guarantee spatial control. First, they use temporal light control.
Meaning time-based.
Yeah. Systems like the PhoBT1 Optogenetic complex are highly reversible. When the blue light goes off, the physical cage aggressively tries to close again.
It actively clamps back down.
It actively disrupts the geometry at the tip of the growing fiber, halting propagation instantly.
Okay, so the lock tries to snap shut. What else?
Second, they use competitive quenching.
Quenching. Like putting out a fire.
Exactly. In the liquid soup, right alongside your building blocks, you introduce inert capping proteins.
Capping proteins.
These are molecules designed to bind perfectly to the growing tip of a cross-beta fiber, but they have no sticky end themselves. They act exactly like PVC end caps.
Oh, so the moment the light turns off and the growth slows down, these capping proteins flood the exposed ends and permanently seal them off.
That is brilliant.
It really is.
And third, they tune the thermodynamic kinetics of the soup itself. By tweaking that hydrogen bond density we talked about earlier, you can design the architecture to be inherently self-limiting.
So it runs out of steam naturally.
The thermodynamic drive of the cascade naturally exhausts itself after adding a certain number of monomers. You completely control the physical parameters of the soup.
So we have achieved full spatial control. We are writing solid, highly conductive, piezoelectric matter into existence using beams of light.
Yes.
Which forces us to ask the next massive logical question for this deep dive. If we can print incredibly complex three-dimensional circuits and wires at the nanometer scale inside a liquid, what kind of computer are we actually building here?
We are building what the framework refers to as the engine of thought.
The engine of thought.
But to grasp how it works, we have to completely discard our current understanding of computer science. If you try to apply traditional binary silicon-based logic to this biological substrate, it makes zero sense.
So no zeros and ones.
No, we're entering the realm of thermodynamic computing.
Okay, let's unpack this. Because in the sources specifically, there's this fascinating internal AI analysis thread. It refers to this as a computational soup. It's a DNA-prion hybrid. Let's look at the two players here. DNA and the prion fibers.
Right. Let's start with the hard drive. Standard silicon computers use binary logic. Zeros and ones, two states.
Yeah.
But DNA has 4 distinct base pairs: adenine, cytosine, thymine, guanine. By using DNA as the memory site, you immediately double the information density per physical operation.
It's a quaternary storage system, base 4 instead of base 2.
Exactly.
But DNA isn't exactly fast. I mean, in nature, reading and writing DNA requires massive, slow enzymes that physically crawl along the strand, unzip it, copy it, and zip it back up.
Right, the biological transcription process.
That takes minutes or hours. You can't run a supercomputer at the speed of biology.
Which is exactly why the framework abandons enzymes entirely for the computation phase. It relies on physical resonance.
Resonance, like sound.
Like electromagnetic waves, specifically terahertz or T-hertz radiation.
Explain how radiation writes data to DNA.
Think of the hydrogen bonds holding the 2 strands of DNA together as tiny microscopic springs. Every single spring has a natural vibrational frequency.
Okay.
The framework proposes using incredibly intense, incredibly short pulses of terahertz radiation tuned to the exact vibrational breathing frequency of the DNA double helix.
So it's like an opera singer hitting the exact right pitch to shatter a wine glass.
Yes, but highly controlled so it doesn't shatter. The T-hertz pulse hits the DNA, the hydrogen bonds violently resonate, and the base pairs unzip and rezip in picoseconds.
Picoseconds, that's a trillionth of a second.
You are reading and writing physical conformational changes at the speed of radiation.
Okay, so the DNA is the hard drive, zipping and unzipping at light speed, and the conductive prion fibers we grew earlier using the light projectors.
They are the logic interconnects. They are the wires connecting the DNA memory nodes, switching their own shapes to perform complex logic operations.
But I have to stop you here. I'm looking at addendum V in the source material, and there is a massive, glaring physical contradiction here.
You're seeing the thermal dissipation objection.
Exactly. Anyone who knows basic physics knows that terahertz radiation is notoriously violently absorbed by liquid water.
That is true.
It doesn't just penetrate harmlessly. It hits water and instantly turns into heat. And DNA only functions in an aqueous environment. It has to be wet.
Right.
So if you blast this microscopic soup with terahertz pulses, the water absorbs the radiation, turns it into heat. Doesn't this entire molecular computer just instantly boil itself alive and scramble its own data?
It is the most critical objection you could possibly raise. But understanding the answer to it actually reveals the greatest strength of this entire architecture.
I'm listening.
What you've just articulated is the classical Silicon Valley view of computing. In classical computing, heat is the ultimate enemy. It is noise. It causes quantum decoherence. It melts chips.
Right. There is a fundamental law in physics called Landauer's principle, which basically says that erasing a single bit of information permanently requires a minimum amount of energy, which is released as heat. The universe literally charges you a thermodynamic tax for forgetting.
That's why Google and Amazon have to build their server farms next to freezing rivers.
Exactly. Traditional computers expend massive, staggering amounts of electricity fighting against the natural thermal chaos of the universe just to keep their data stable.
But the framework introduces a 2025 breakthrough in thermodynamic computing, citing work from Caltech and the Ludwig Maximilians Universität in Munich.
Okay, what's the breakthrough?
In this biological architecture, heat isn't destroying the computation. The heat is the battery.
I'm going to need you to walk me through that carefully. How does heat act as a battery for calculation?
Because the physical computation is designed to be thermodynamically favored. In a silicon chip, you are forcing electrons into unnatural, high-energy states to represent a 1 or a 0.
They don't want to be there.
Exactly, they desperately want to escape. But in thermodynamic computing, the system is physically engineered so that the correct logical answer corresponds to the lowest possible free energy state of the molecular network.
Wait, so the molecules naturally want to settle into the correct answer.
Exactly. Imagine a large, bumpy physical landscape, like an egg carton.
Okay, an egg carton.
And you drop a marble into it. The marble represents the current state of the computer. The correct answer to your equation is the deepest divot in the carton.
So gravity just pulls the marble into the deepest divot.
Right. But the marble might get stuck in a shallow divot on the way down.
Yes, local minimums.
So what do you do to get it unstuck? You shake the carton. That shaking is thermal fluctuation. It is Brownian motion. The heat generated by the terahertz absorption, that random, chaotic jiggling of water molecules, literally jolts the system.
It shakes the egg carton.
Yes, it helps the physical molecular states slide down the energetic hill until they settle into the deepest, most stable valley.
Which is the correct logical state. That is unbelievable. The T-Earth's pulse provides the initial shock, kicking the system out of equilibrium. The resulting heat cascade actually stabilizes and propagates that state.
The thermal chaos is literally doing the mathematical heavy lifting.
You aren't fighting the heat, you are surfing it.
The heat drives the system toward equilibrium, and equilibrium is the computational result.
For you listening to this is a total conceptual inversion. We are looking at a computer that operates near the absolute thermodynamic floor of physics.
It doesn't fight nature's chaos. It harnesses it as an engine.
The mismatch cost, the wasted energy between what computation theoretically requires and what our current clumsy machines use, effectively drops to zero.
Because the system preserves information through geometric equilibrium rather than by constantly erasing and overwriting states through brute force voltage.
Okay, my mind is thoroughly expanded on computing. But let's ground this again. Computing, structure, and wires are only 3 pillars of a civilization.
Right, you need more.
If we're genuinely talking about building a self-sustaining, planetary-scale technological civilization from the ground up, using biology, we need baseline power.
And massive amounts of it.
Massive, continuous, reliable baseline power. You can't run a global grid on the ambient vibration of a room. Which brings us to Section 5, unlimited power and the prion nuclear convergence.
This is where it gets heavy.
How on earth does a strand of protein fundamentally rewrite nuclear energy?
To appreciate the solution, we really have to look at the flaw in current nuclear battery technology, specifically what we call beta voltaics.
Beta voltaics, not like a massive nuclear reactor with cooling towers, but a small battery that uses radiation.
Right. In a standard beta voltaic cell, you have a chunk of a radioactive isotope sitting physically adjacent to a semiconductor transducer.
Okay.
The isotope naturally decays over time. When an atom decays, it shoots out a high-energy particle, like a beta electron.
And that particle hits the semiconductor.
Eventually, it has to travel out of the radioactive chunk across a physical gap and hit the semiconductor. When it hits, it knocks electrons loose, generating a current.
And I'm guessing that journey across the gap is highly inefficient.
It is incredibly wasteful. It suffers from a physics problem called self-absorption.
Self-absorption.
The emitted beta particles lose huge amounts of their kinetic energy simply crashing into other radioactive atoms inside the chunk before they even make it to the surface to cross the gap.
Oh, they just bounce around inside their own material.
Right. Because of this, current beta voltaics struggle to reach even a 1% to 3% conversion efficiency. Most of the decay energy is lost as useless internal heat.
But the framework cites a massive leap forward in 2024.
Yes, a benchmark study from Soochow University involving a coalescent transducer.
Coalescent, meaning they completely combine the radioactive material and the solar panel-like transducer into one single thing.
They combine them at the molecular scale. They synthesized a material where the radioactive isotope and the transducing atoms were interspersed perfectly in the exact same crystalline lattice.
So there's no gap.
Zero gap. Because they were in such intimate atomic proximity, the self-absorption gap was totally eliminated. The decay event literally happens inside the conversion medium.
So every single bit of energy from the atom splitting hits the transducer instantly.
Exactly. Every recoiling atom, every phonon, every particle couples directly to the material. The result was an 8,000-fold efficiency gain over traditional beta voltaics.
8,000 times. And Enfield takes this 2024 coalescent breakthrough and applies it directly to the prion architecture.
He does. He points to the existence of engineered superurinal binding proteins, or SUPs.
Okay, superurinal binding proteins.
These are synthetic proteins that have been designed by computational biologists to bind to dissolved uranium ions with what is called femtomolar affinity.
Stop there. Femtomolar affinity. What does that actually mean in plain English for us?
A mole is a standard unit of measurement for molecules. Femto means 1/4,000,000th.
Okay, so unbelievably small.
If a protein has femtomolar affinity for uranium, it means its molecular claw is so perfectly shaped and chemically so aggressively attracted to uranium that if you put just a few atoms of uranium into a massive Olympic swimming pool of water,
The protein will find it.
this protein will instantly find them and lock onto them with unbreakable strength.
Unbelievable precision. So what does Enfield propose we do with these SUPs?
He proposes using CRISPR to insert these super uranium binding claws directly into the amino acid sequence of our SUP35 yeast prion.
Let me put this together. You take a massive bioreactor vat, you fill it with seawater, which naturally contains trace amounts of dissolved uranium.
Or you use wastewater from an old nuclear plant.
Its built-in claws naturally pluck the uranium atoms out of the water and lock them into its own physical core at exact, evenly spaced atomic precision.
The fiber becomes a biomineralized lattice. You have naturally occurring radioisotopes locked in angstrom-scale proximity to the highly conductive, highly piezoelectric amyloid core.
The battery isn't a lead box holding a radioactive core. The battery is the core. The very wire you use to build the structure is itself a continuous nuclear reactor.
We must be incredibly precise with our terminology here to avoid panic.
Right, fair enough.
It is not a reactor. It does not sustain a critical, controlled fission chain reaction. Nothing is going to melt down like Chernobyl.
Okay, good to clarify.
It is a multimodal, solid-state nuclear energy converter. It passively captures energy from natural, spontaneous radioactive decay.
But it does so with terrifying efficiency.
Because it harvests that single decay event through 3 simultaneous channels.
Trimodal harvesting. Break those 3 channels down for us. How does it squeeze every drop of juice out of one atom decaying?
Channel 1 is the beta-voltaic channel. When the uranium atom decays, it shoots out an alpha or beta particle. Because the atom is locked directly adjacent to the tyrosine-rich conductive core of the fiber, that high-energy particle immediately tears through the pi-p stacked rings. It knocks electrons loose and generates an instant, direct electrical current.
Okay, direct particle hit. Standard beta voltaic. What is channel 2?
Channel 2 is the piezoelectric channel. This relies on the conservation of momentum. When a gun fires a bullet forward, the gun violently recoils backward into your shoulder.
Yeah.
When a uranium atom fires an alpha particle, the atom itself violently recoils.
It physically kicks backwards.
Yes. That violent atomic kick creates a microscopic mechanical shock wave, phonon, and that ripples violently through the surrounding crystalline lattice.
But remember what our lattice is made of.
Exactly.
The amyloid scaffold.
Yeah.
You already established that the amyloid scaffold is massively piezoelectric.
Yes, so when the atom recoils and shakes the fiber physically squeezes its own asymmetrical lattice. It converts that mechanical kinetic shock wave directly into a second spike of voltage.
That is just brilliant. Conventional nuclear batteries lose all of that kinetic recoil energy as useless vibrational heat. The prion architecture harvests the physical shock of the atom splitting.
It catches the recoil. And channel 3.
Yeah, what's the third one?
The thermal gradient channel. Even with the first 2 channels harvesting energy, the continuous decay still generates localized heat.
Yep.
Because these fibers are only 10 nanometers wide, the decay creates a massive, sharp temperature difference between the burning hot inside of the fiber and the cooler fluid on its surface.
Oh, a temperature delta.
The architecture acts as a nanoscale thermoelectric generator, converting that temperature gradient into a third stream of electricity.
Pre-harvesting channels from a single atomic decay event. Operating simultaneously across trillions of nanoscale generators, woven seamlessly into the very fabric of your buildings.
It's pure efficiency.
But I have to push back here, hard.
Go ahead.
If we are mass-producing biological nuclear batteries, growing them in vats, using them as structural girders, weaving them into our power grids, aren't we just creating a massively distributed, completely uncontainable radioactive hazard?
It's the obvious concern.
What happens if an earthquake hits and a building made of this stuff shatters? Are we just dusting the city in radioactive uranium dust?
It is the ethical and safety nightmare that traditional nuclear engineers bring up immediately. But the framework addresses the structural containment realities directly.
No.
We have to look back at the mechanical properties we established in Section 1.
The extreme toughness and indestructibility.
Exactly. The amyloid scaffold is essentially indestructible. More importantly, it is naturally radiation hardened.
Right, the flexible bonds.
When gamma rays hit rigid crystalline silicon or metal, they knock atoms out of place, causing the containment vessel to become brittle and eventually fracture over decades.
It's a great.
But the cross-beta architecture is held together by those flexible, dynamic hydrogen bonds. The radiation passes through, or the bonds briefly break and instantly snap back into place due to thermodynamic favorability.
It is a self-healing containment vessel.
It functions as the ultimate solid-state encapsulation matrix. It is actually magnitudes safer than traditional heavy steel and concrete containments.
Because it flexes.
Because the uranium atoms are chemically locked into a matrix that can survive being boiled in acid, bombarded with gamma rays, or subjected to massive mechanical trauma without ever releasing its payload.
And if it does take microscopic damage from an extreme impact, The self-replicating nature of the prion cascade just recruits new protein from the environment and naturally seals the crack.
It's an autonomously self-healing nuclear battery.
That is insane.
Yeah.
Okay, we really need to zoom out now. We've solved the structural brick. It's tougher than steel. We've solved wiring and sensors using intrinsic conductivity and piezoelectricity. We've solved manufacturing using optogenetic printing. We've solved computing using the thermal chaos of nature. And we've just solved unlimited baseline power using coalescent nuclear biominerals.
It's a complete toolkit.
When you put all of those pieces on the board at the exact same time, how does this redefine what it means to be a technological civilization?
We enter the realm of holographic information geometry.
Holographic information geometry.
Okay, to truly understand the scale of what Enfield is proposing, we have to touch on a concept by the legendary theoretical physicist John Archibald Wheeler.
Wheeler, the physicist who actually coined the term black hole, right?
Yes. In his later years, Wheeler proposed a concept called it from bit.
It from bit.
His proposition was that physical reality at its most fundamental quantum level isn't actually made of matter or energy. It is made of information.
Like binary code.
A binary choice. The answer to a yes or no question. Geometry emerges from information, and physical matter emerges from geometry.
It from bit. Everything is just data rendering itself as reality.
Exactly. Now, let's apply that to the Bekenstein-Hawking entropy formula. This is the formula that describes the thermodynamics of a black hole.
Okay. It's here with you.
It states something profoundly counterintuitive. The total amount of information contained inside the three-dimensional volume of a black hole is actually encoded entirely on its two-dimensional surface, its event horizon.
Hold on. Let's make sure that's clear. You're saying the 3D stuff inside the black hole doesn't really matter. All the instructions, all the data that makes up that volume is just painted on the 2D shell around it.
Yes. Think of a security hologram on a credit card. It's a flat, two-dimensional piece of foil, right?
Right.
But it contains all the information necessary to project a fully three-dimensional image. The 3D volume is just a projection of the 2D boundary.
Okay, I follow the physics, but how does black hole entropy connect to a vat of growing protein?
The framework makes its boldest, most astonishing claim right here. It argues that the cross-beta amyloid scaffold operates on the exact same physical principle. The fibril is a literal physical hologram.
Explain that. Because in a hologram, if you cut it in half, you don't get half the image. Every tiny piece of the film contains the complete image of the whole.
Exactly. And the same is true for the prion fiber. Think about the growing tip of the fibril.
The end of the thread.
It is a two-dimensional surface. But every single microscopic fragment of that surface geometry, no matter how small, contains the complete, perfect architectural blueprint to nucleate and regrow the entire three-dimensional structure.
The part contains the whole.
The surface geometry of the fibril tip is the instruction manual.
It is the instruction, the memory, and the physical product simultaneously.
Yes, it requires no external factory. It requires no robotic assembly arms. It operates entirely on holographic information geometry.
So the amino acid sequence encodes the 2D fold, the fold encodes the surface geometry, and the surface geometry just pulls molecules out of the liquid to physically write the 3D material into reality.
You aren't building a material anymore. You are writing a physical geometric program that autonomously executes itself into solid matter.
Which fundamentally rewrites our understanding of the Kardashev scale.
Entirely.
Right, the Kardashev scale.
Yeah.
For those who aren't huge sci-fi nerds, this is the system astrophysicists use to rank hypothetical alien civilizations based on how much energy they can consume.
Right, type 1, 2, and 3.
A Type I civilization controls all the energy of its home planet. Type 2 controls the total output of its host star. Type 3 controls the energy of an entire galaxy.
And how do we normally picture a type 3 civilization?
Giant metal megastructures.
Oh.
Fleets of mining ships tearing apart asteroids, massive robotic foundries smelting titanium, spherical cages built around stars made of quadrillions of tons of steel.
Right, and that assumption, that infrastructure is a giant dead machine separate from the biological civilization that uses it, is the exact industrial revolution mindset that this framework shatters.
Because of it from it.
If we connect it from bit to the prion architecture, the framework proposes what Enfield calls the type 3 node. In a prion logic framework, you do not need a planet-sized industrial base of steel mills and silicon fabrication plants to expand across the galaxy.
Why not?
because the factory is a single microorganism. The entire technology stack, the structural manufacturing, the DNA computers, the optogenetic light printers, the nuclear energy harvesters, all of it can be genetically encoded into the DNA of a single microscopic cyanobacterial seed package.
So you don't launch a massive mining fleet, you launch a biological probe the size of a coffee can.
Or smaller.
It drifts across the galaxy and lands on a dead, carbon-rich asteroid in another solar system.
And the moment it lands, it uses ambient starlight for energy, and it consumes the local carbon, nitrogen, and trace metals. It begins to divide.
It uses biological exponential replication rates to grow.
It doesn't build infrastructure. It becomes the infrastructure. Within a few decades, that single microscopic seed package has used local chemistry to autonomously grow a fully capable, technologically advanced civilization from scratch.
And what exactly does it grow into around that star?
The logical thermodynamic conclusion is a piezoelectric Dyson swarm.
A Dyson swarm is usually a trillion giant solar panels orbiting a star to capture all its light. But you're saying the swarm is grown organically.
Yes. Vast orbital clouds of engineered cyanobacteria continuously extruding thousands of miles of amyloid-based piezoelectric membranes, which are automatically metallized by the asteroid's own trace elements.
My God.
And because these biological membranes are deeply piezoelectric, they don't just harvest the photons of light. They harvest the violent mechanical stress of the solar winds slamming into them. They harvest the gravitational tidal stress of the star itself.
They harvest the physical stretching of space.
Yeah.
And because these amyloid membranes are inherently conductive wires, the entire biological swarm acts as a massive unified electromagnetic array.
Yes.
The source material mentions using this array to actively manage the star's fusion rate via acoustic resonance.
Exactly. By pulsing synchronized electromagnetic fields, the swarm can induce acoustic sound waves deep within the plasma of the star.
It's like playing the star like an instrument, using incredibly precise sound waves and magnetic pressure to physically squeeze the stellar core, throttling the star's fusion energy output up or down depending on what the swarm needs.
A biologically managed stellar-scale tokamak reactor?
That is just hard to even picture.
A civilization built on this architecture doesn't wait passively for stellar evolution to swallow their planet. They actively manage their star's lifespan and output.
The biology is the prerequisite. Once you have the holographic protein architecture, everything else is just inevitable thermodynamic arithmetic.
That's the core of the thesis.
Okay, we have to synthesize this journey for everyone because the scale of this is honestly dizzying. We started this deep dive looking down a microscope at a single highly misunderstood biological fold, the cross-beta architecture.
Yep, the mad cow protein.
We unpacked how manipulating its hydrogen bonds makes it tougher than spider silk and impervious to acid. We learned how stacking tyrosine rings lets it conduct electricity intrinsically.
And how manipulating its asymmetrical lattice allows it to harvest electrical power from the physical shock wave of a decaying uranium atom.
And then we explored how we can tame that wild exponential growth using optogenetics, literally projecting solid matter into existence using structured light.
Right. We saw how the random chaotic heat of terahertz radiation bouncing off water molecules can act as the battery for a non-binary DNA computer.
We moved from a nanoscale structural trick to a self-replicating scalar-scale civilization built entirely out of programmable self-healing protein.
That's quite a leap.
It is. So for you, the listener, the absolute core takeaway from Enfield's framework is that the hard lines we currently draw between hardware, software, and biology are entirely artificial, and they're rapidly dissolving.
What we currently consider cutting-edge human achievement, like our multi-billion-dollar silicon fabrication plants, our lithium-ion battery gigafactories, or massive titanium rocket hulls, it might soon look like banging heavy rocks together in the mud.
Compared to the elegance of thermodynamics-driven biological programming.
We are looking at the transition from extracting our civilization from the Earth to growing it seamlessly from pure information.
Which leaves me with one final, lingering, slightly unnerving question. Something to chew on that isn't explicitly mapped out in these equations.
Oh.
If a civilization actually achieves this, if they eventually write their entire infrastructure, their global power grid, their supercomputers, and their interstellar spacecraft into the very DNA of their own biology, What happens to the concept of waste?
That's a good question.
What happens to obsolescence? I mean, if your towering buildings, your computational machines, and your own biological body are all made of the exact same programmable, self-repairing, light-written protein, does technology cease to be a tool that you pick up and use?
Wow.
Does it instead become the environment that you simply are?
That is the ultimate civilizational paradigm shift, when the tool and the creator become completely indistinguishable.
We will leave you to mull that one over. Thank you for taking the deep dive with us today.
So today we're going to dive into a technology that's, well, it's pretty mind-bending. We're going to start with one of biology's most feared villains and end up with what could be the blueprint for a whole new kind of civilization. It's a wild ride based on the work of Zachary David Enfield, and it basically reframes everything we thought we knew about the very building blocks of life. Our story starts in a pretty dark place, with the prion protein. For decades, right, this is what we've known it as, the thing behind these awful neurodegenerative diseases. It's this misfolded, infectious protein that's so tough it can survive things that would totally obliterate almost any other form of life. It is, for all intents and purposes, indestructible.
But what if that indestructibility isn't a bug, but a feature? What if it's actually a clue? And that's the big question that just flips the whole script. What if the very same logic that makes a prion so dangerous is also the key to creating the most advanced materials we could ever imagine?
To really get that, we've got to look at this villain's secret identity, because hiding underneath all that scary pathology is an architectural principle that has staggering power and, honestly, a kind of elegance to it. You see, it's a tale of two folds. On one side, yeah, you've got the pathological prion that we're all afraid of. But on the other side, you have these things called functional amyloids. Now, these proteins use the exact same structural logic, but not to cause disease. They use it to create materials with unbelievable strength and stability in nature. So it's not the protein itself that's good or bad, it's all about the way it folds. And this is the secret sauce. It's called the cross-beta architecture. Just try to picture it. You've got these protein strands layered up, all perpendicular to a central axis, and they're all locked together by this super dense network of hydrogen bonds. The plywood analogy here? It's perfect. It really is like nanoscale plywood. And that's what gives these materials their incredible combo of stiffness and toughness.
Okay, so keeping that principle in mind, let's look at how this thing becomes truly revolutionary. Because now we're moving past just biology and into a whole new class of engineering material.
Okay, now when you look at this chart, the key insight isn't that amyloid beats steel in raw numbers, because it doesn't, not yet anyway. The absolutely mind-blowing thing here is that it's already playing in the same league as other supermaterials. I mean, look at its performance compared to spider silk, which is kind of the gold standard for biological toughness. And it does all this while being a fraction of the weight of steel. So this material really has this whole package of superpowers. It's tougher than spider silk, it's incredibly light, and get this, it can self-replicate. That means you can literally grow it in a bioreactor instead of forging it in some massive factory. And remember that whole indestructibility thing? Yeah, it's resistant to heat, acid, even gamma radiation, conditions that would just destroy our current electronics. For something like aerospace or deep-sea tech, this is an absolute game changer. So we have this miracle material, great, but how do you control it, right? How do you actually shape it into something useful? Well, the answer is a process that sounds like it's straight out of science fiction, prion optogenetic lithography. Let me break down how this works. It's so cool. First, you engineer a protein that really wants to form these amyloid structures, but you put it in a cage that's light sensitive. So in the dark, nothing happens, it just sits there. Then you shine a really precise pattern of light into the solution, kind of like a tiny projector. And wherever that light hits, it unlocks the cage. That's the trigger for the self-assembly to start. And it just grows from there. You are literally printing a 3D structure using nothing but light. And what could we print? Oh my man, the possibilities are just staggering. Think about it. Spacecraft hulls that can heal their own cracks in the vacuum of space. Or 3D printing food that molecularly replicates the fibrous texture of a steak. Growing biological circuits that act as conductive wires. You could even print medical scaffolds directly inside a wound to help guide tissue regeneration, all of it programmed by light. This is where we go to the next level, a level of scale that is almost impossible to imagine. We're not just talking about printing objects anymore. We're talking about printing the actual infrastructure of a civilization. Let's start with just a number, 8,000. According to the research Enfield cites, that's the potential boost in efficiency for nuclear energy if you can build the power source at a molecular level. So instead of some big, clunky battery, what if the material was the battery? It's about making the power source and the thing converting the power one and the same. And this is where that prion architecture becomes so critical. By engineering these amyloid fibers so they can literally grab onto and bind with nuclear isotopes, you create a material that is its own nuclear battery. It can harvest decay energy in 3 ways, all at once. It can capture electrons directly, it can convert the physical vibrations from decay into electricity, and it can harvest the heat. It's a self-assembling, multimodal power source. Just wow. So we have a material that we can shape with light, and that can power itself. You can see how this starts to lead to some really profound, almost philosophical thoughts on what this technology actually is. Enfield makes the case that this isn't just like a hologram. In a very real sense, it is one. Think about it for a second. A tiny little piece of a holographic plate has all the information for the entire image, right? Well, in the same way, a tiny little fragment of this amyloid fiber has the complete blueprint to replicate the entire structure. It means the information needed to build a complex 3D world is encoded right there on a simple 2D surface. It's this incredible link between biology and the fundamental laws of physics. So what's the roadmap look like? I mean, this is ambitious, for sure. But within the decade, we could be looking at industrial production and the first biosensors. Then by the 2030s, that could scale up to aerospace testing, maybe even pilot programs for food printers. Mid-century, that's where it gets really futuristic, with things like self-repairing hulls on deep space vehicles. And the long-term vision, we're talking beyond 2060, is a fully integrated self-replicating material platform on a planetary scale. And that really brings us to the final, and probably the most critical thought from Enfield's work. This isn't just a new material. This is a whole new paradigm, where the lines between machine and computer and organism they start to dissolve. And as we stand on the edge of being able to wield this kind of power, the ultimate question really becomes one of wisdom and understanding. What does it really mean to hold a technology that can literally write itself into existence?